Today, we're diving into a really cutting-edge approach to fighting cancer. What if, instead of just attacking it from the outside, we could train our own immune cells to become a precision army? We're going to look at how a very special type of cell, a true general in our body's defenses, can be harnessed to create a personalized, powerful vaccine against tumors.
Okay, so here's the game plan. First, we're going to meet the key player in all of this. Then, we'll see how scientists pick the best ones for the job, how they give them a specific target, and finally, we'll get into the really revolutionary stuff, the future of how these vaccines are being built.
All right, first up, let's meet the absolute star of the show, the dendritic cell, or DC for short. These guys are the masterminds, the generals, who are just critical for kicking off a powerful, targeted immune attack against anything that shouldn't be in our bodies.
So here's the most important thing you need to know about them. They're messengers. Think of them like intelligence officers out on the front lines. They find the enemy, in this case, a cancer cell. They gather intel on it, and then they race back to the command center to brief the special forces, which are our T cells.
And this is what makes them so cool. Dendritic cells are basically a bridge between two different worlds of our immune system. They take information from our body's general alarm system, the one that just kind of reacts to everything, and they deliver it to the highly specialized, highly trained forces. They're the ones who turn a vague threat alert into a precise seek and destroy mission.
The best way to think about an immature DC is as a patrolling officer. It's stationed right at your body's borders, your skin, your lungs, your gut, all the places that are in direct contact with the outside world, just waiting for trouble. So its mission plays out in 4 key stages. First, it patrols, using all these sensors to detect signs of danger. Once it finds something, it engages, it literally swallows the invader and chops it up into little pieces called antigens. This whole process triggers a transformation. The DC matures, it stops patrolling, and it starts traveling. Its destination, the nearest lymph node, which is basically the immune system's military base, to go brief the troops. And this is the briefing. Inside the lymph node, which is just packed with T cells, the DC presents those little enemy fragments. It holds them up on special molecular platters called MHCs. Think of it like holding up a wanted poster. But, and this is so important, it's not enough to just show the poster. The DC also provides a second signal using molecules like CD80 and CD86 that basically shouts, "This is real, this is a threat, time to act, now."
Okay, so we've met the general, but now for the next step. You see, when you're building an elite army, you have to be really picky about who you recruit. Not all dendritic cells are cut out for this mission.
And right away, we hit a pretty big snag. As you can see from this chart, the best dendritic cells, the natural-born killers, are incredibly rare. They make up maybe 1 or 2% of our blood cells. On the other hand, you have these things called monocytes, and we have tons of those.
This table really lays out the dilemma. For a long time, scientists worked with what are called MoDCs. They could grow these in a lab from those plentiful monocytes, which was great, easy to make. But they had a huge weakness. They were bad at traveling to the lymph nodes, and they were missing a key skill called cross-presentation. The rare, natural blood DCs, though, they're experts at both.
So let's meet the core team. First up, the CDC1s, or the killer's trainer. Their special skill is something called cross-presentation. This is a game changer. It lets them take bits of an external threat, like a virus or a cancer cell, and show it to your CD8+ killer T cells, the assassins of your immune system. Then you've got the CDC2s, the general's advisor. They're in charge of activating the CD4+ helper T cells, who are basically the generals that orchestrate the entire adaptive response. And finally, the PDCs, the antiviral sentries. When these guys sense a virus, they unleash a tidal wave of powerful antiviral signals called type 1 interferons to put the entire body on high alert. And that one skill, cross-presentation, is everything. It's the unique ability to show intel to our most lethal soldiers, the CD8+ T cells, and give them the order to kill. It's like showing them a mugshot of the exact enemy to hunt down. The lab-grown MoDCs just aren't good at this, but the natural blood DCs are pros. That makes them the perfect recruit, if you can find them. So let's say we've found our elite recruit. Now what? Well, you have to give them a mission, a crystal clear target. How exactly do we train these dendritic cells to go after cancer and only cancer? Well, it all comes down to specificity. You see, early on, scientists used targets that were common on cancer cells, but they could also be found on healthy cells. Not ideal. And cancer could just mutate to hide them. But then came neoantigens. These are a total game changer. They're unique fingerprints created by the cancer's own mutations, meaning they only exist on tumor cells. It's a target the cancer literally cannot hide. So how do we find them? It's basically high-tech genetic detective work. Scientists take a piece of a patient's tumor, sequence its DNA, and use powerful software to find those unique mutations. Then, they actually synthesize these little molecular mugshots in the lab. These are what get loaded onto our dendritic cells to train them for their mission. All right, let's put all these pieces together and see how this works in what's called an autologous vaccine. And that's just a fancy way of saying from you for you. It's a completely personalized therapy built from the ground up for one single person. So picture this. The whole process is custom built. Your own blood is drawn. Those super rare dendritic cells are carefully isolated. They're trained in the lab using the unique fingerprints from your specific tumor. And then this bespoke, personalized vaccine is injected right back into you. But, and this is a huge but, as incredible as this sounds, it's got some massive real-world problems. For one, it's ridiculously expensive. It also takes weeks to create just one dose, and the logistics of doing this for every single patient are just a nightmare. So it's really, really hard to scale up. Which brings us to the big question. How do we get the incredible power of a dendritic cell vaccine without the crushing burden of personalizing it every single time? And this, my friends, is where the story gets really, really exciting. I mean, what if we could find a way to make a powerful, effective dendritic cell vaccine that was just ready to go? Something sitting on a shelf that could be given to lots of different patients. Well, the answer might just lie in a special subtype of dendritic cell called a plasmacytoid DC, or PDC. Scientists have actually figured out how to grow a stable, endlessly replicating line of these cells in a lab. This creates a universal foundation for a vaccine that could be mass-produced. The advantages here are just huge. You can make it at an industrial scale, which brings the cost way down. You can treat tons of patients with a standardized product. And get this, because of their unique properties, the patient's body doesn't reject them. That means you can give people booster shots to keep their immune response firing on all cylinders. But it's not just about making it easier or cheaper. These off-the-shelf PDCs might actually be, well, supercharged. Studies are showing they can activate way more of those critical killer T cells. Plus, they pump out massive amounts of a substance called interferon, which is like a battle cry that rallies the entire immune system to the fight. And the early results we're seeing from this whole neoantigen approach are just so inspiring. Take this one trial for liver cancer. They saw an 84.6% survival rate without recurrence after 1 year. Now, that was a personalized vaccine, but it shows you the raw power of this strategy. Now, trials for the new off-the-shelf versions are getting started for lung cancer and other tumors, and there's a ton of hope. This quote from one of the researchers really nails it. Whether it's a completely custom-built vaccine or a powerful off-the-shelf version, the big idea is the same. We're using nature's own messengers to teach our body how to defeat its most complex enemy. And that leaves us with one final and really crucial thought. As this amazing science moves forward, the challenge changes. The off-the-shelf approach is one potential answer, but we have to keep asking ourselves, how do we make sure these revolutionary, life-saving therapies are actually available to everyone who needs them?